And when I am copying a DNA molecule, if I were to start at one end, let's say with the primase, and I'm starting and I'm copying all the way down, well, the leading strand, I can copy all the way, but the lagging strand is going to, the lagging strand can copy all the way, but the leading strand is going to end up having that little piece of primer that was part of the original molecule that cannot easily be cut out. So that little piece of primer is going to end up being what they call a telomer. And that is a little piece of DNA that is not copied. Now this does not, this is something that is not really a problem for a long time. So in other words, cells can copy their DNA over and over and over again, but every time they do it, they're cutting off a little piece of the DNA. It's kind of like if I had paragraphs of important information, and before that paragraph I had a whole group of thes, or I had a whole group of ands, some nonsense word, or a word that doesn't really have anything to do with the paragraph. And let's say I have thousands of those. I can literally cut off one each time I do the replication process, and it's not really cutting into my paragraph, which is my important information. So this is why the telomeres can keep being cut off time after time, but this is also why cells age, because at a certain point they will be copied at so many times that the telomeres are now cutting into the paragraph, the important information. And when they start cutting into the paragraph is when the cells really cannot be replicated just like the last cell. In other words, the DNA is then compromised. So this is, set in obsolescence for our bodies. This is why we age. And these telomeres are constantly happening, and the cells that do the most mitosis, like in your skin, your hair, those places are the places you see the telomeres being cut off the earliest, right? So in reproductive cells, they have an enzyme called telomerase that will copy the telomere. So this is only reproductive cells. So telomerase, and it will copy that telomere. And this is happening in cells that are made for being passed on to the next individual. It's good that the telomere is not cut off so that everybody starts with a full set. And that telomerase is only found in our gonads. That would be in our ovaries and testes. Now, interestingly, telomerase is also found in cancer cells. So this is why cancer will do mitosis and make tumors that keep growing over and over and over again. Because normally cells are limited in their distribution, or in their growth, that is, they're limited in how many times they can do mitosis by the neighboring cells. They have being squished in with a neighbor cell will make it stop doing mitosis until one cell dies, or one cell is like ripped out and you get a cut or something like that. And it will replace it, but otherwise cells are limited usually by their neighbors. In the case of a cancer cell with the telomerase, it is going to keep doing mitosis until it's making a tumor, and these tumors are just taking up space, which is why cancer is painful, because it's taking up space that it shouldn't normally take up. Anyway, so that just the process then of replication, just to review, includes the initiation protein. Sorry, I'll hold it up here. The initiation protein. It will include the helicase, which opens up the molecule. It will include the single-strand binding proteins that are holding it apart, primase that's laying down the primer, and DNA polymerase that is copying. Make sure that you understand the difference between the leading and lagging strands. All right, thank you.